So today's video is going to be slightly different. So far, we've been reviewing a lot of concepts from Unit 1, but in this episode, we're going to be covering how to apply those concepts to FRQ as you may see them on your exam or on the May exam. So stick around. Hi everyone, my name is Mikey from AVO Prep Academy, and today we have a very special episode. I've created 4 questions that are very similar to the types of questions that you may see on College Board's exam or on your school test as well. So we're going to take a look at those and we're going to solve through them using the concepts that we've been discussing in the previous 4 videos. If you haven't seen those videos, I will also link all those links in the description below so you can check them out as well. Now, before we get started, the FRQ section of the AP Biology exam can be very challenging. The free response question requires not just your ability to recall knowledge based on priming, but to bring them out based on your organization of ideas within your mind. And today we're also going to talk a little bit about how to approach those FRQ questions. And in fact, I'm going to do that right now. So free response questions are challenging because you have to be able to synthesize a lot of different ideas from many different units and combine them together in its application to the question at hand. However, there is one way to approach FRQ that's going to help you, and I call it the ABC method. So what is the ABC method? Well, A, answer, B, background, and C, connect. Well, in AP Bio FRQ, they're going to give you guys questions that say describe some phenomenon or explain some phenomena or identify something. And in each of these questions, there is going to be a definitive answer. So we want to make sure that we put that forward so that the readers know that you know the answer. So in the very first part of ABC, the A stands for answer. So answering the question with the most concise sentence that you could develop is going to be important. But B, which stands for background, requires you to then tie together some of the more fundamental concepts that you've learned in AP Biology into this question. And as you do this, what you're basically showing the readers is that you know your stuff. It's an opportunity for you to showcase your knowledge on the relevant course content that pertains to the question at hand. And C, the last part, is about connecting that background to the very specific question being asked, which then loops all the way around to the answer that you've already given in A. So this answer, background, and connect is always going to work, but remember that in some questions you might have to provide a couple of sentences for the background or even a few sentences for connections. So just remember that ABC does not necessarily mean 1, 2, 3 sentences, but they may span across a paragraph, and that's kind of what you want to do for some of the more long-form questions as well. Now that we have the general FRQ strategy out of the way, let's go ahead and take a look at these questions that I've developed for Unit 1. So Unit 1 was all about biochemistry, which was primarily on water, carbon, macromolecules. Okay, so that's chapter 3, 4, and 5. So let's take a look at how those concepts can be synthesized into FRQ style questions. So the way that we're going to do this is that I've brought over my iPad so that we can take a look at the screen together and you can follow along in addressing these questions. But keep in mind that these are full-length questions, which means that this video might be best viewed on your desktop or a tablet. So if you haven't done so, click the like button so later on, if you need to switch devices, you can easily find this video. Okay, so let's take a look at my iPad and get started with today's main focus. So over here, what we see is a seemingly challenging question with a lot of chemistry, but let's take a look at what the question says first. A particular membrane-bound receptor protein is composed of 3 polypeptides. Each of the polypeptides occupy the following region of the protein: extracellular region, lipid bilayer, and the intracellular region. The following is a short sequence of polypeptides from the polypeptide in the extracellular region. And then we have this image of a polypeptide, and then we have a rare mutation causes the following changes to the polypeptide sequence described above. And here we have that altered polypeptide sequence. All right, guys, so before we get started, let me just explain a couple of things here. They tell us something about this protein having 3 polypeptides, and that means that there are 3 tertiary structures that have combined together into a quaternary structure. So that's going to be important probably because they specifically told you that, or I did because I made these questions. Now, let's take a look at what else it says. It says each of the polypeptides occupy the following region of the protein. We have the extracellular region, we have the lipid bilayer, and then we have the intracellular region. And what I've done is I've actually gone ahead and drawn out this protein, and you can see right here that we do have the extracellular region of the protein that I'm coloring in green, and then the intracellular, which I'm also going to color in green, and we're also going to have this lipid bilayer region, which I'm going to draw in purple. So the reason that I'm doing that is that lipid bilayer is in fact a non-polar region, and as you guys might remember, extracellular is aqueous and the intracellular is aqueous, meaning that this cell is surrounded by water and its cytoplasm is also comprised primarily of water. So then let's take a look at this short polypeptide sequence and see what we can say about this. Okay, so I want to pick a nice color here. Let's go with this bright green. In an earlier video, I did mention that you don't need to memorize all of those 20 amino acids, but I did say that you should be able to look at its structure and be able to know some behaviors about that particular amino acid. And this is going to be our practical application of that. Because if we take a look, we can tell that we have a peptide bond there, another peptide bond here, another peptide bond right here, meaning that we have 4 amino acids in this polypeptide. And the R groups I'm going to outline right here, another one right here, another one right there, and the last one right there. So those are going to be our R groups, and everything else is our backbone. Now, in order to identify the properties, we're looking for very specific chemical signatures. So for instance, in this very first amino acid that I'll call number 1, we have a hydroxyl group, which means that this number 1 amino acid is most likely polar. Number 2, another hydroxyl group, probably polar. Number 3, seems like an acid because it has a negative charge. That's also going to be polar, as in it dissolves in water. And then here we have another hydroxide group, meaning that this is also polar. So what we're looking at right here is a chain of amino acids which all dissolve in water, and they're all hydrophilic amino acids. However, let's take a look at this mutation, because once we get down to this polypeptide, we have some major differences. Once again, let's identify where those peptide bonds are and where those R groups are. Right there, we have another one, we have another one, and we have another one. Now, it seems to me that the first and the last amino acids are the same, meaning that number 1 and number 4 are still polar. However, if you take a look at number 2, that's a methyl group, a hydrocarbon. Number 3, methyl groups, more hydrocarbons. So 2 and 3 are going to be our nonpolar amino acids. Now this is going to be very reminiscent of that hemoglobin example where we had glutamic acid, which then mutated into valine, going from polar to nonpolar, meaning that there was going to be a structural problem, and that's probably what we can deduce about this type of mutation as well. So let's go ahead and take a look at the answer choices, and let's see if we can also apply that ABC methodology here a little bit, and I'll probably put up the text on the screen as I explain the answers. Number 1, identify the 3 regions, extracellular, lipid bilayer, and intracellular, as either polar or a nonpolar environment. Now this one is relatively easy because I've already drawn what the answer should be right here below the questions. However, if we do want to use that A to B to C methodology, let's just briefly talk about how we might do that. So A is going to be the direct answer. So I might say something like, the extracellular region is polar, the lipid bilayer is nonpolar, and the intracellular region would be polar as well. Okay, so that addresses the A. What about B, the background? So let's talk about that. We can say something like, in biology, cells reside in aqueous solutions. So the outer portions of the cell will be a water-based environment. Also, the cytoplasm of the cell, which is the intracellular environment, is also some 70 to 80% water, making it polar. However, the lipid bilayer is comprised of phospholipids, meaning that the lipid tails that face each other in that bilayer are going to be non-polar. So there we go. So that's the background. Okay, so now let's connect it. We might say something like, because of these facts, extracellular regions which faces water would be polar, this intracellular region which contains the cytoplasm would be polar, while the lipid bilayer with those hydrocarbon tails would be non-polar. Okay, so that's how we address questions like that. So now let's take a look at number 2. It says, describe how the mutation to the polypeptide in the extracellular region causes a loss of function in the receptor protein. Okay, so the loss of function, what does that mean? Well, that means that the protein is no longer going to be able to do what it was supposed to do, which was essentially to receive signals from the outside and transduce it to the inside of the cell. Now, because it says describe, what we need to do is, again, follow that A, B, C methodology, keeping in mind what's happening here with these mutations that cause nonpolar amino acids to take the place of the original polar amino acids. So here is the most direct answer. The mutation to the polypeptide, which resulted in two nonpolar amino acids taking place of two polar amino acids, would cause a loss of function in the receptor protein because the structure of the polypeptide would be different as a result of the changes to its primary sequence. Okay, so that is the most direct answer. Now let's give them our background. Protein structures are created from the sequence of amino acids described as the primary sequence. The amino acids along a chain with its R groups will interact and thus fold into a three-dimensional conformation or a shape that allows a function to arise. However, as the amino acid sequence changes, the chemical interactions between the R group changes, resulting in a different structure. These different structures may lead to an altered form of a protein, which may have reduced or no function at all. Okay, so that about sums up the general background of proteins. So how do we connect it to this question at hand? Well, we might say something like this. Because this protein is a receptor protein, if those nonpolar amino acids are now in the position where those polar amino acids were supposed to be, the changes to the active site of that protein may result in its inability to connect to either a signaling molecule or a molecule within the cytoplasm, which is responsible for connecting that signal to the cell. Therefore, the function of this original receptor protein would no longer work because of the changes to its structure. Okay, so that's how we sort of round everything up together in questions like number 2. Now let's take a look at number 3. It says quaternary structures of proteins also involve many chemical interactions, including ionic bonding, covalent bonding, hydrophobic interactions, and hydrogen bonding. If a toxin that interrupts covalent bonding between sulfur atoms was added to a solution containing the receptor proteins described above, identify which level of the protein structure would be compromised due to the toxin and explain why. So at this point, guys, I'm not going to do the whole A, B, and C because I think that's just going to eat up a ton of time. I'm going to be putting up an answer key as well to the link description below, so you can obviously download it and take a look at my answers too. However, let's take a look at what this question is asking. So if we have a protein, you guys have learned that we have the primary, we have the secondary, we have the tertiary, and we have the quaternary structures. And in the question, we're also told that the quaternary structure can involve covalent bonding. Now, where else is covalent bonding found? Well, it's found in that tertiary structure as well. And this type of bond is actually very specific. If you remember, there is an amino acid called cysteine with a sulfhydryl group. And what happens when two sulfhydryl groups combine together is that an H2 gas is removed, and we have what we call the disulfide bridge, which is a very strong bond that holds the structure of both tertiary and quaternary structures together. So if they're asking which level of the protein structure would be compromised, that would certainly be the tertiary and the quaternary, and the reason for this is going to be on account of this amino acid cysteine, which forms disulfide bridges. Okay, great. So that is how we solve a long form question. Now we have a couple of other questions that we need to be looking at, so let's go ahead and take a look at the second question. This one is a little bit simpler. It says cellulose is a polysaccharide composed of beta-glucose monomers. The enzyme amylase found in animals cannot break down beta linkages in cellulose. As a result, certain animals, such as herbivores and termites, form symbiotic relationships with microbes and protists in order to digest cellulose. Briefly explain why amylase can digest starch but not cellulose. So guys, when we take a look at this question, it's really asking, why does this protein do one thing but not other things? So this protein, which is our enzyme, specifically is going to be amylase. Well, it's going to perform its function on account of the fact that it has a particular structure. And the structure of amylase is going to allow its interaction with starch, which is then going to break down those alpha glucose linkages in that starch. However, cellulose, with its beta linkages, with beta glucose monomers, well, they're going to be shaped differently. If you look at the picture of the alpha glucose versus beta glucose polymers, you'll notice how the hydroxyl groups are not in the same place between these two molecules. And as a result, amylase is only going to be able to interact with one, but not the other. Okay, so simple enough. Let's take a look at another one, shall we? Okay, so in this question, it says a scientist investigating the role of certain macromolecule in a cell decides to design an experiment to identify whether the macromolecule is a protein or a nucleic acid. In the experiment, the scientists decide to use radioisotopes of certain atoms to track and identify the macromolecule in question. After the completion of the experiment, the scientist identifies the macromolecule as emitting radioactive decay particles from phosphorus but not sulfur. Based on these results, what is the identity of the macromolecule? So in order to address this question, you have to first identify those 4 macromolecules. We have carbohydrates, we have lipids, we have proteins, and we have nucleic acids. Okay, so once we have these 4 macromolecules, you need to know what the elemental compositions are for each one of these macromolecules. So what that means is carbohydrates are going to have carbon, hydrogen, oxygen, and all the way over here I'm going to write nitrogen because chitin does have nitrogen, but you probably don't want to think about that. Okay, so lipids. What about lipids? We have carbon, hydrogen, oxygen, and phosphorus. The reason for that is, of course, our phospholipids. Proteins? Well, we have carbon, hydrogen, oxygen, nitrogen, and sulfur. Sulfur for that cysteine, which is, again, that special amino acid that we talked about just now. Methionine also has it too, but of course, nitrogen from those amino groups. Nucleic acid has carbon, hydrogen, oxygen, nitrogen, and phosphorus. That phosphorus being the phosphate backbone, nitrogen for the nitrogenous bases, and of course carbon, hydrogen, and oxygen are found just like all the other macromolecules here as well. Now, the reason that this is important is that the whole sulfur versus phosphorus difference between proteins and nucleic acids is in fact how we discovered that DNA was responsible for genetic information storage way back in the day. So if we have radioactive decay from phosphorus, that would be nucleic acids as the answer for this question. Let's take a look at the last one. I like this last question. It shows you a graph of DNA denaturation. What does that mean? Well, in DNA denaturation, as temperature is slowly brought up, DNA strands can unwind and become single-stranded from its original duplex. Now, this happens as a result of thermal energy compromising those hydrogen bonds between the bases, and that is the focus of this question. So let's take a look. It says, the following diagram shows the denaturation of 2 DNA molecules, red and green, under varying temperatures. Guys, be careful. Each one of these red and green are double-stranded DNA, all right? So it's not like we have the red and the green strands, which are comprised as a single DNA. These are 2 separate double helical molecules. And it says the Tm represents the temperature at which 1/2 of the DNA duplex is denatured to single-stranded DNA molecules. All right, good. So we have this graph here, and there's the Tm for the red, and then there's the Tm for the green. And it says, when DNA denatures, its 2 strands unwind to become single strands rather than incurring backbone breakage and fragmenting into multiple double helix pieces. Explain why the DNA denatures through dissociation rather than fragmentation. So guys, this is a really great question. And the reason is that it's really asking about what types of bonds hold together the deoxyribonucleic acid. Because if I were to draw DNA like this, what they're asking is why is it that they fall apart like so instead of breaking up into broken pieces of DNA. So here we need to understand that the backbone of DNA are created from what we call phosphodiester bonds. Phosphodiester bonds, which are covalent bonds between the phosphates and the ribosugars that run down the length of the DNA backbone. However, the connection between the bases that are holding the 2 strands together are hydrogen bonds, and that is a much weaker bond. In fact, it's not technically a bond. It's an intermolecular interaction. So as that temperature goes up and the structures get compromised, the first things to go are, of course, the weaker bonds, the hydrogen bonds, rather than the breakage of covalent phosphodiester linkages. So that about answers number 1. Now, number 2 says, given the graph above, identify and explain which of the 2 DNA molecules have greater adenine-thymine content. So let's first talk about adenine and thymine versus guanine and cytosine. Because if you remember from an earlier lecture, you might remember that adenine and thymine are connected via 2 hydrogen bonds, while guanine and cytosine are connected by 3 hydrogen bonds, which means that if a DNA molecule has greater AT content versus a molecule with greater GC content, the DNA containing the AT content is going to be weaker in holding the two strands together. And the GC heavy DNA will be stronger because of greater hydrogen bonds, or greater number of hydrogen bonds rather. Okay, so let's take a look at this graph and see if we can figure it out. Well, there's one way to do this. You can see that half of the DNA of the red DNA strand is denatured at around, let's say, 76.5 degrees, while half of the DNA strand with the green one is denatured at 80, let's say 4 degrees, which means that DNA that's red instead of the green denatures at much lower temperature than the DNA that's labeled in green. And what that means is it's probably weaker. Now, the other way to do this is to simply pick an arbitrary temperature like 80 degrees and see how much of that DNA is denatured for red versus green. At 80 degrees, that DNA is denatured at a much greater rate here than they are here. Now, don't worry about this OD260. The idea is that the y-axis is going to tell you the degree of denaturation. So this red strand of DNA must have more AT content because it's denatured halfway at an earlier temperature, or at any given temperature, it's more denatured than the green DNA. Alright guys, so that about sums up our video today. I know that there were only 4 questions, but remember on the real AP exam, you only have 6 FRQ, so it's not that bad. However, for this unit test, I hope that these questions help you out a little bit. And if you have a teacher who likes to create really tricky questions, well, you might see something that's very similar to what we've discussed today. So hopefully that was very helpful to you guys. If you haven't done so already, subscribe to our channel and like these videos because we're going to be doing this for each of the 8 units within the next year. So I'm going to try to keep pace with your school's curriculum. I may be a couple of weeks ahead or behind, but as long as you stay on top of our videos, I think you'll do awesome in your AP Biology course as well as the exam. So my name is Mikey. It's been great having you guys here today. We'll see you guys in the next video. Take care.